What's up guys? Welcome to my channel Absolute Biopharma. I am your host Pallavi, and on this channel I bring you the content related to the biopharma industry, various job vacancies, interview questions, trainings, technical content, and the list goes on. And if you are new to this channel, you first happen to click on this video randomly, or maybe YouTube suggested this video to you, you might want to consider subscribing because I bring to you the most relevant content related to the biopharma world. So be the part of this journey and hit the subscribe button, hit the bell notification so every time I produce a video, you will get notified. So without any further ado, let's get started. So today's topic is about monoclonal antibodies. Before starting this video, if you want to know what are antibodies, I have already made a video on the antibodies. You can watch the video and have a look about antibodies by clicking on the I button. So in this video, I am going to cover what are therapeutic antibodies or monoclonal antibodies, what are the types and suffix used to denote the maps, production of antibodies by hybridoma technology, production of antibodies by recombinant DNA technology, why we prefer monoclonal antibodies, their clinical applications, and some of the US FDA approved human monoclonal antibodies. So let's begin the video. Let's start with what are therapeutic antibodies, or you can say MAPs. So these are the type of proteins that are made in laboratory and can bind to certain targets in the body, such as antigens on the surface of the cancer cells. So there are many types of monoclonal antibodies, and each mAb is made so that it binds to only one antigen. So these are being used in the diagnosis and treatment of many diseases like cancer. They can be used alone or to carry drugs, toxins and radioactive substances directly to the cancer cells. So MAPs are the warriors inside our body. They are the part of our immune system, recognizing and fighting against the foreign invaders, against antigens. So they bind to a broad range of antigens and are used for medical and diagnostic applications. So let us see how they are made and what they are made up of. So there are four types of monoclonal antibodies. First is murine, chimeric, humanized, human. So let's begin with the first antibody, that is murine antibody. So the origin of these murine antibody is from the rodent host. Patients treated with murine antibody develop an allergic reaction to these molecules termed as human anti-mouse or anti-murine antibody, that is HAMA response. So this response is known to cause quick elimination of these molecules from the human and therefore limits the therapeutic application. So murine antibodies are often engineered to reduce their natural immunogenicity towards human patients. So genetic manipulation of antibody first generated chimeric antibody structure containing the mouse Fab region and human FC region. So the combination of mouse Fab region and human FC region with different origin allowed to increase the compatibility of these mouse generated antibodies toward human cell. These consist of an antibody's original antigen binding variable domain with a constant domain from different species. So example of chimeric antibody is rituximab. So example is rituximab. So it is the first drug that is approved by FDA for the treatment of cancer. So it is one of the most successful anti-cancer biotherapeutics around the globe. So beyond clinic, this antibody has proven to be the useful tool for immunoassay development such as ELISA. So these humanized antibodies are made up of small part of mouse proteins that are attached to the human proteins. Last is the human antibody. These are made up of fully human proteins. Let's understand the type of antibodies by figure. So first is murine antibody. This is the structure of antibody. Here is the variable region. Here is the constant region. So this is the light chain and this is the heavy chain. So in murine antibody, it is completely derived from the mouse. So here there will be no change in the variable region or the constant region. So for murine antibody, the suffix used is OMAB. So for chimeric antibody, XmAb is added. like infliximab, rituximab. For humanized antibodies, zumab is added, palivizumab, and fully human antibody mubab is added. So if you see in the figure, in murine antibody there is 0% contribution of the human antibody. In chimeric there is 65% human antibody. In humanized it is 90%, and in fully humanized it is 100% human antibody. So here generic suffix used is omab for murine, zmab for chimeric, zumab for humanized, and umab for fully humanized. So if you see the potential for immunogenicity, it will be high in murine, and if we go towards the fully human, it will be low. Next comes the production of the MABs. So there are many methods by which monoclonal antibodies can be produced. First is mouse, then phage display technology, transgenic mouse technology, single B cell, and then transgenic animals. So let us understand each production by figure. So in mouse hybridoma technology, here is a mouse that will be immunized with targets. Then the splenocytes are harvested, which generates the hybridomas. Screening will be done. Then the mouse antibody will be produced. So these antibody can be chimerized. Then these can be humanized. Phage display technology. In this phage displayed antibody libraries will be there. From that, biopanning will be done with the targets. Again the screening will be done. Then the IgG antibody construction will be done, and then human antibody is produced. MAP production by transgenic mouse. First we have to immunize the target or the mouse. Then splenocytes are harvested, which will generate the hybridomas. Again the screening will be done, and human monoclonal antibodies are produced. Last is by the single B cells. So human will be there. So here the primary bone marrow cells will be there. The B cells are sorted out and they are labeled with the antigens. Then again the screening part will be done, and by PCR variable and constant regions are constructed and ligated later on. So in hybridoma technology, the fusion of antibodies producing spleen cells from the immunized mice or rats with immortal myeloma cells is done. So in hybridoma technology, there is a fusion of two types of cells. First is antibody producing spleen cells, and next is immortal myeloma or cancerous cell line. Here is a figure that will give you a clear understanding of hybridoma technology. So first the mouse is immunized by an antigen, then it is fused with B cells and myeloma cells, then in the hat selection they are grown. Screening is done by ELISA, then hybridoma screening is done for antibody of interest. It means that whichever antibody is of our interest, that will be screened out. After screening, the antibody of interest are produced by hybridoma clones. Later on, the monoclonal antibodies are expanded and stocked. So let us see a quick procedure of antibody production. So in first immunization of mice and isolation of splenocytes. So mice are immunized with an antigen and later their blood is screened for antibody production. So the antibody producing splenocytes are then isolated from in vitro hybridoma production. Preparation of myeloma cells. So myeloma cells are immobilized cells that are once fused with the spleen cells and will result in the hybridoma capable of unlimited growth. Myeloma cells are prepared by fusion. Next step is fusion. In fusion, myeloma cells and isolated splenocytes are fused together to form hybridomas in the presence of polyethylene glycol, which causes cell membranes to fuse. So clone screening and picking. In this, clones are screened and selected on the basis of antigen specificity and immunoglobulin class. So in functional characterization, the clones are confirmed, validated and characterized by ELISA, each having potentially high productivity. Scale up and weaning is done. The scale up is done for the desired antibodies, and weaning is done by selecting agent. And the expansion is done in the bioreactor or the large flask. What is the drawback of hybridoma technology? It has insufficient immune response to highly toxic or conserved antigens. So here a point is there to be noted. All the antibodies which are currently in clinical development are of human origin or at least humanized in some aspect to prevent immunogenicity. So consequently, transgenic animals, especially mice, have been developed which contain human immunoglobulin gene, reported solving the problem of immunogenicity but not the need of an efficient immune response after immunization. So finally, in vitro selection technologies such as antibody fast display or ribosomal display provide a solution for generation of human antibodies. So let us see how MABs are produced by B cells and specially targeted antigens. So in this technology, cell lines are genetically modified such that the immunoglobulin IgG genes were inserted into the genome, replacing the endogenous IgG genes and making these animals capable of synthesizing fully humanized antibodies upon immunization. So here is a figure. In first step, there will be an immunization of animal, and then in second step, the fusion will be there of plasma cells and myeloma cells. Assay will be there for culturing supernatant. Then the sequencing will be done by molecular biology. Then cloning of the MAP sequence is done by expressing vector. Production and purification of the recombinant monoclonal antibody is done. So if you see in the figure, the mice is there or an animal is there, it is immunized by antibodies. So here the plasmocytes and the myeloma cells are fused. Here the screening is done by the ELISA plate. So here the determination of MAB is done by DNA sequencing methods. So molecular cloning is done by fusing various variable heavy chain, variable light chains. Then the recombinant antibody is constructed. So MABs, if we see, are highly specific, which means they can recognize and bind to a single antigen binding site. This means they are targeted to a specific antigen and is a key trait that makes them useful in diagnosing and treating disease. So MABs have applications in the research field, diagnostic field, therapy. They are used in standard assays like immunoblots, flow cytometry, immunohistochemistry, and they are useful in production of monoclonal antibodies. So let us see some of the clinical applications. So therapeutic antibodies are currently approved for disease treatment. So MABs are increasingly used for broad range of targets, like they are used in the field of oncology, they are used in immunology, they are used in hematology. So here are some of the cancers where the MABs are used as drugs. First is lymphoma, myeloma, melanoma, glioblastoma, neuroblastoma, sarcoma, colorectal cancer, lung, breast cancer, ovarian and head and neck cancer. So other applications, if you see, number of target proteins known to function as their stimulatory or inhibitory checkpoints of immune system and targeting the programmed cell death protein 1. So if this kind of activity is there for this type of programmed cell death PD1, nivolumab and pembrolizumab are used. For ligand programmed death, following MABs are used: durvalumab, avelumab and atezolizumab. So next, for cytotoxic T lymphocyte associated antigen, ipilimumab are used. So let us see some of the US FDA approved MABs. So here is the MAB, then the brand name, the company who produced the following MAB, the target where it acts, what is the type of the antibody, which technology is used to produce the antibody, for what it is used, and when the US approval is given. So rituximab, Mabthera is the brand name, Rituxan is there. Who produced it? Biogen, Roche and Genentech. Then the target, it acts on CD20 gene. It is produced by Chimera. So the format is chimeric IgG1. It is produced by hybridoma technology and it is used for non-Hodgkin lymphoma, and the approval was given in 1997. So, as I have mentioned before, also, this is the first drug that is used for cancer, and it is the first drug that got the US FDA approval. So, rest are pelvizumab, infliximab, trastuzumab, alemtuzumab, adalimumab, then ibritumumab, omalizumab, cetuximab. So you can have a quick look on the following antibodies. Next, bevacizumab, natalizumab, panitumumab, ranibizumab, ustekinumab, canakinumab, golimumab, belimumab, pertuzumab, trastuzumab, nivolumab, pembrolizumab. So here what I have mentioned are some of the drugs. There are many drugs outside which are having US FDA approval. So thank you so much for watching my video, and I hope it added some value to your knowledge. And if you like the video, then don't forget to subscribe, like and share my content. So I will see you in my next video that will be on bioreactor series. In that you will get everything related to the bioreactors. So till then, have a great day.